Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO, and your host. Today, Dr. Denise Cai will present How Does a Fearful Experience Alter Our Past Memories? BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 183 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has had such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded more than $430 million to fund more than 6,200 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Denise Cai. Dr. Cai is Associate Professor in the Department of Neuroscience and the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai. She was a 2018 Young Investigator and a 2021 Honorable Mention for the Friedman Award. Our webinar will begin with Dr. Cai's presentation, which will then be followed by question and answers. To submit your questions, please use the questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now I'm pleased to introduce Dr. Cai. Denise, the floor is yours.
Awesome. Thank you so much, Jeffrey, and thank you for that wonderful introduction. And thank you so much to Brain and Behavior Research Foundation, who invested in us early on when this was kind of a crazy project and high risk. But because of your generous funding and all of the support, we've been able to make tremendous strides in answering the question of how does a fearful experience alter our past memories? So I'm really excited to share the update with you guys. And thank you everyone for joining today. I really appreciate that creating the space and giving us your time. So my lab, I'm a memory researcher and I study memory in mice. And my lab is really interested in how is it that our brain is able to accumulate what seems like an infinite number of memories across our lifetime. Somehow our brain knows where to store the memory so that we can later retrieve it for accurate recall. And I would argue that, I think memory research is one of the most important areas to research because the accumulation of all these memories across a lifetime, I believe, is what defines our human experience. And just as an example, my daughter, she's only 8, but she's had so many different experiences already in her life. And somehow her brain knows where to store this and how to retrieve it. And so this is a question of interest in our lab that we model in mice. Now, we're also particularly interested in how is it that aversive memories are stored in the brain? And why do we care about aversive memories or how trauma memories are stored in the brain? I probably don't have to tell this audience that much, but it's really important because the majority of the population will experience a traumatic event at one point in their lives. So some examples, we often think about military trauma, sexual assault, child abuse, vehicular accidents, natural disasters, and really over the last couple of years, we've all collectively been experiencing the trauma of the global pandemic of COVID-19. And the list of trauma goes on and on. And the consequences of psychiatric function are widespread after trauma. And we know that trauma can lead to post-traumatic stress disorder that is quite debilitating. It can potentiate anxiety and depression, as well as substance dependence. So understanding how our brain processes trauma is really important as we think about how to develop prevention and interventions for these mental health challenges. Now, why is mental health research important to me? If you don't mind me just giving you a little bit of a personal perspective on this. So this is my family. These are my parents, my husband, my son. You saw my daughter in earlier pictures. And my parents are immigrants. They survived the communist revolution in China. Coming to the United States, they've also had challenging times here as immigrants. And for as long as I can remember, my mom has really struggled with anxiety, but undiagnosed. And many times she would have these panic attacks and she's like, I have heart problems. She would go to the hospital again and again, not understanding why she kept having these attacks. And my dad would always say, oh, it's nothing. It's just in your head. And after I had the privilege of studying psychology for my degree in college, I was able to convince my parents that, Dad, you're right. It is in Mom's head. It's called her brain. And this is a brain condition, and she needs to go see brain doctors. And after seeing a really excellent team of psychiatrists, she was able to get medication. And my mom wanted me to share the story, and on her behalf and our family's behalf, really thanks so much of the scientists, the preclinical scientists, the clinical scientists, all of the healthcare workers for developing these important life-saving medications so that she can go on and live a wonderful life. And since she's found these medications that's worked for her, she hasn't had panic attacks. And now she's one of the very lucky ones because there's so many people in which are resistant to treatment, meaning that medications are not working for them. And so we need to continue to work hard to better understand, brain challenges and how to improve mental health. And certainly we could not have done this without the generous support of taxpayer money, as well as generous donors who contributed to so much of the innovative research that have led to successful interventions and treatments. So on behalf of my family, we just want to say thank you to all the scientists and the funders. Okay, but I'm a memory researcher, okay? So we want to ask, well, how are memories stored in the brain? So from a lot of research over many decades, we've learned that memories are stored in sparse neural populations across the brain. And here is just a cartoon of cells in the brain that contain the memory. And we've learned that when memories are recalled, many of the same cells are being reactivated. And this is thought to underlie the stable representation of a memory in the brain. And when a different memory is recalled, when a different memory is recalled, different cells in the brain are being reactivated, and this is how the brain knows. In this case, for example, my daughter's at the dentist versus climbing a tree. And so different memories are stored in different neural ensembles in the brain. As I told you, I'm a mouse researcher. So how do we actually visualize memories in the brain or cells in the brain? So let me just take a minute to show you the technology that my collaborators and I developed, and this is the open source miniature microscope. This microscope on the left-hand side is no bigger than the size of my thumb. It's only about 3 grams, and we can put it on a mouse's head and peer into the awake and active brain as animals are freely behaving, interacting with the world, creating memories, and we can see their cells light up in real time, as you can see in the video on the right. So using this technology and other technologies, we can visualize brain activity while animals are exploring the world. And I just want to take 1 minute to tell you a little bit about this project of developing this technology in the open source platform. And this was a project that started while I was a postdoc at UCLA under the leadership of Alcino Silva and Payman Golshani. And I worked directly with Daniel Haroni, who is a postdoc as well, who was the lead tech developer, and Tristan Schumann, who is a lead surgical person. And together we developed this open source system. And when we developed it originally, we thought, okay, if there's some way that we can make it so it's super cheap, maybe 10 labs around the world might be interested in using this to answer questions about brain and behavior. And since we released the first version in 2016 and second wire-free version in 2020, over 500 labs around the world have been using the scope, and over 2,000 mini scopes have been built. And so this has really inspired a revolution in open source neural technology in being able to innovate and share openly with labs around the world, so we can all answer questions in a more innovative and make more progress faster. Okay, so back to memories, right? So as I said, I don't study humans, I study mice. So how do we see memories in the brain? So here's an example. We'll put a mouse in a box. It's a brand new environment, and then we give the animals a foot shock. And we know that memories of this contextual representation and paired with the shock is represented in neural ensembles that are included in the brain regions like hippocampus, which is important for storing memory, and amygdala, which is important for storing aversive memory. And when we put the animals back in the box, we know that many of the same cells are being reactivated, supporting the long-term memory. And behaviorally, we can see that the mouse remembers being in the box because now they're not shocked in the box, but they show behaviors such as freezing, which is a natural defensive behavior indicating that they remember being shocked in the box despite not getting shocked anymore. And when they are put in a different box, different cells represent this red context, and they also don't express freezing in this box. And so this is just an example of how, as an animal researcher, we are visualizing memories in the brain and correlating with this behavioral output. Okay, so if it's really these neural ensembles that underlie the stable storage of these memories, can we erase these aversive memories, much like the movies, like Men in Black, for example? So scientists have developed really cool tricks to be able to do this. And what they do is they tag the cells, in here the blue cells, that are active during the initial encoding. When they put the animals back in the box, the animals recall that memory. Again, many of the same cells are reactivated. And now when they put the animals back in the box, they can shine light using this tool called optogenetics, in which when shining light, we can silence specifically these cells that were previously active, and it decreases the memory, such as now the mouse does not freeze in this box anymore. So a number of studies have shown that we can selectively inhibit, either with light or with drugs, these specific neural ensembles and erase specific memories. Therapeutically, this is very cool, right? Because if you have a trauma memory, wouldn't it be nice just to erase a trauma memory without erasing the memory of that great first date with your husband, or maybe the last conversation you had with your mom before she passed? So if we want to manipulate trauma memories, we want to make sure that we're being very selective and we're not just erasing all memories. And so a lot of this research helps us think about therapeutic interventions for how the brain processes trauma. So in addition to erasing memory, what about artificially activating these memories? So the same logic applies here. If that this is the neural ensemble that really underlies the storage of the memory, if we put the animal in a different box, can we just artificially activate the memory of that fearful experience and induce the animal to recall that fearful experience and then lead to the animal freezing? And indeed, a number of researchers have shown that just by selectively artificially activating these neural ensembles that were active during the memory encoding, indeed they can induce, at least partially, this behavioral recall of that memory. So altogether, a lot of research has gone on to show that memories encoded in sparse neural ensembles spread across the brain, and that these neural ensembles can be manipulated to either suppress or activate these memories. And we can think about their therapeutic implications. Now, these are for how individual memories are processed. And a lot of work in the field has really nailed down how are individual memories processed in the brain. But as I had previously shown you, memories are not formed in isolation. You and I, we're constantly experiencing new things all the time. Our brain is constantly accumulating memories. And so how does the memory of this conversation or this seminar affect your future learning of other things? Or how does this update what you knew about memories in the brain, right? How do we stably represent memories while flexibly updating and integrating new information so that we can move around in a dynamic world? And specifically today, and the question that I had proposed in my Brain and Behavior Research Foundation grant a few years ago, as Jeff mentioned, is really to ask, how are aversive memories linked with other memories? So to give you an intuition of why this question might be important, here is an example. So here's a picture of my kids. They are going to the dentist. This is a pretty aversive memory, right? Because they found out they had cavities and they had to get it drilled, right? Pretty aversive. And based on the literature, we know that this memory is probably encoded in sparse neural ensemble distributed across the brain. Now, if my kids were smart, it would really behoove them if they can think back in time, or if the brain was able to search back in time and ask, why do they have so many cavities? And they might think back to when their dad made them all these sugary raspberry gimlets. Don't worry, this is sans alcohol, kid approved. And if they could somehow link these two memories across time, then linking the present to the past might be able to predict future outcomes so that they can make better decisions in the future, such as drinking less sugary drinks or at least brushing their teeth after and flossing. So to model this in mice, what we do is we put an animal in a box and boom, we give them a strong foot shock. And again, we know that this memory is encoded in sparse neural ensemble. And we ask, now, do they link this memory to a novel experience that precedes this? And we call this retrospective linking. And do they transfer the fear of this aversive experience to the safe context, which they saw maybe days ago, despite never being shocked in this red box? Is there some kind of updating mechanism such that it links the present to the past? Alternatively, it's possible that after an aversive experience, the animals link this prospectively to future novel contexts, and this fear is transferred across days to other experiences in time. So to test this, Joe Zaki in the lab with Denise Morales-Rodriguez and Taylor Francisco modeled this behaviorally. So in this experiment, there's two groups. There's the group testing prospective linking, and then there's a group testing retrospective linking. So the prospective linking group is the animals get shocked in the blue box. 2 days later, they're brought back to the red box, and we're going to ask, do they transfer the fear of that blue box to that red box, despite never being shocked in that red box? Alternatively, animals might transfer the fear of the blue box in the retrospective group to that red box seen 2 days prior, despite never getting shocked in that red box. Okay, so before we test for the transfer of fear, let's first make sure that our manipulation worked, that indeed shocking in that blue box was aversive, and that when we put them back in that blue box, they remember being shocked in that blue box by showing freezing behavior, which is their natural defensive behavior, which is what you see here. And here what we show is that their percent of time they spent freezing in that box, about 50%, and is no different in the prospective group or the retrospective group. Okay, cool. So now we're asking our question, do they transfer the fear prospectively or retrospectively? So in the prospective group, what we do is now we split the animals, half of them get tested in this neutral context, right, they've seen before, and we ask them, do they transfer the fear to that box? We also test them in a novel context because we want to know, well, are they just scared of everything? Maybe it's not just specific fear transferring to the red box, but it's fear just transferring to any new box. So what we see here in the prospective group is that you see much less freezing in the neutral group than in that blue bar, and no different than a novel group, suggesting that while they remember being shocked in that blue box, they don't transfer the fear to that neutral box in the red or the novel box in the green. Okay, what about the retrospective group? So here we're asking, do they transfer the fear from this blue box to the red box seen 2 days ago? And indeed they do. So selectively they show that there is higher freezing in this red box despite never being shocked in there. and that this transfer fear is selective, as we don't see very much fear in the novel box. So for the rest of the talk, we're going to focus on this retrospective linking. And what we're hoped to be modeling is kind of, this example I gave, that after an aversive experience, how does the brain link it back to past experience to link the two to make predictions about the future? Okay, so for the rest of the talk, I'm going to go between some cartoons to show you our kind of framework for it, and then some data. And if you get a little lost in some of the graphs, I know some of you guys are real hardcore, want to see the data, want to see the graphs. Don't worry, I will summarize everything in the cartoons. So the question now is, okay, we see that after an aversive experience, a fear is transferred to a neutral event from 2 days ago. But how does the brain do this? So based upon what we know from the literature, we know that after exploring in a novel environment that's safe, the memory is encoded in this neural ensemble. 2 days later, when they go experience this aversive experience, it's encoded in a separate neural ensemble. And what we also know from the literature, what I have not told you yet, is that after learning this aversive experience, or really any new experience, during some offline period, a lot of this is just animals back in the home cage, while the quiet rest, while they're sleeping, there is reactivation of the recent experience. And this is typically thought of as neuronal replay. And this replay, or this reactivation of the neural ensemble of what just occurred, is important for consolidating or stabilizing that memory, such that that memory can be converted into a long-term memory. Now, what's really interesting, so this is full circle to my graduate work, because my graduate work was actually on sleep and memory. And what we showed was that sleep was not only important for reactivating and stabilizing recent events, but sleep was also really important for integrating what you just learned with your past knowledge. And so we hypothesized that maybe after this aversive experience during this offline period, it's not only that recent experience of that blue box that is being reactivated, but that maybe your brain is searching for past events to reactivate as well to link the two. Interestingly, work from Andre Fenton's lab showed that the higher the stress of an event, the further back in time you can pharmacologically manipulate other memories that were encoded days ago. And so this led to our hypothesis that maybe the more stressful this aversive experience is, the more likely your brain is going to reactivate past memories to try to find what is that missing link to that maybe predicts that future aversive experience. And that by this reactivation of both the aversive blue box and this neutral red box, but by reactivation of both of them, this might link the two, such that when animals go back to the neutral context, this context in which they've never been shocked, that it might also trigger the memory of the blue box, which would induce fear or freezing as the behavioral output in these animals. So to test this, Joe Zaki in the lab, a very talented graduate student who's shared by Kanika Rajan, who is also a BBRF fellow scholar, what he did was he used calcium imaging. So again, this is similar to the tool I showed you before, where we put this miniature microscope on top of the animal's head, and we peer into the brain of the hippocampus, and he's recording hippocampal activity while animals are exploring two different contexts. Here I'm showing you two different groups. And the difference between the two groups is that in the low shock group, when animals enter the blue box, they get a baby shock, where in the high shock group, when animals enter the blue box, they get a very high shock. And then after the animals are done with this aversive experience, Joe puts these animals back into their home cage and records their neural activity for an hour. And then he looks at all the cells that fire during this offline reactivation while the animals are in their home cage, right? So they're no longer in these boxes anymore. And he says, okay, every cell that he sees, he will sort them into one of four groups. So if a cell from the low shock, for example, was previously active when the animal was exploring the red box, then he would categorize this as part of ensemble A. If the cell that was active during this offline period was also previously active in ensemble B, he would call this ensemble B. If the cell was active in both A and B previously, he would call this part of ensemble A and B. And then most of the cells, about 70% of the cells that he saw during this offline period, were not previously reactivated, so he called this a non-ensemble. Okay, so what did he find in the reactivation of the hippocampus during the low shock? Okay, so let me take you step by step through this. So what we would expect is that cells that were active during the blue box, so would be likely to be reactivated during their home cage experience. And this would replicate what we've seen, what I refer to as neural replay or memory reactivation, and that's really important for stabilizing the memory of the blue box. And that is indicated, so on the y-axis is the activity rate, so how much does the cell fire. And what you can see is that cells that were active in the blue box, indicated by both the blue and purple lines, are much more likely to be active compared to the gray line, which are the non-ensemble neurons. And this activity decreases over the hour, once again, replicating what other people have found. Interestingly, the memory ensemble for this red box seen 2 days ago is much lower than the activity of the memory ensemble for the blue box. That's also not surprising at all, and what you would expect, because that memory was from 2 days ago. There's going to be, excuse me, less reactivation now. Okay, but the question is, what happens after a high shock? And our hypothesis was that after a really aversive experience, the brain doesn't only reactivate this neural ensemble of the blue box, but the brain is searching for past events to link the memories together, and that it would reactivate the ensemble of the red box as well. This is precisely what we see. The main takeaway here is that now that red line jumps up and is no different than the blue and purple line. And so that this suggests that increasing high shock is increasing the reactivation of memories from 2 days ago. And so, okay, so this is really interesting. If that's true, here's a cartoon of what we found, which is that after this aversive experience during the offline period, not only is the ensemble of the blue box reactivated, but the ensemble of the red box is also reactivated. So we wanted to ask, if this activity during this offline period is important, what if we just inhibited the activity? What if we silence these cells just during this offline period? And then when we put them back in that red context, do they unlink the two? And, you know, therapeutically, this is really important, right? So far, I've pitched out this is a really adaptive process to link aversive experience to past predictors. But you can imagine that this can become maladaptive if there's too much linking across too many experiences that are unrelated. Okay, so in this experiment, what Zach did was he is trying to, or he successfully decreases activity during this offline period. And what he's using here is chemogenetics, and this is a specific DREADS system in which we can inhibit the activity of hippocampus by injecting drugs. So in this experiment, what Zach does, Zach is a very talented postdoc in the lab who's been working on a number of these experiments. So what Zach has done is in two groups. So the GFP group is just the control group. We inject GFP in the brain, but no active HM4DI. And then another group is the active HM4DI, which we inject into the hippocampus. And here Zach exposed animals to two different contexts separated by 2 days, and all animals get a shock in the second context. Now, after that, he puts the animal back in the home cage and he gives them this drug, CNO, in which in the group with the HM4DI, it will decrease activity of hippocampus while animals are in the home cage. Now he behaviorally tests this when he brings the animal back, and he asks, when he put the animals back in the neutral context, do they still transfer that fear from that blue box to that red box? And how selective is it? Because now he can also test for generalization to a novel context. So consistent with data I've already shown you in the control group, the GFP group, what Zach sees is that indeed the animals transfer the fear from the blue box to the red box, as shown by the high level freezing in that red box. Again, just reminding you, despite never being shocked in this red box. Also important to note is that this transfer of fear is quite selective, as you don't see animals freezing very much in this novel context. again, suggesting that it's not just that the animals are more sensitized to fear in general, right? This transfer fear is quite selective. So what happens in the group where he tries to decrease activity during this offline period? Well, what he finds is he gets rid of selective transfer of fear, but it also looks like there's an increase in generalization, right? And maybe we were not that surprised by this, because what I told you before is that to stabilize this memory and consolidate the memory of this blue box, the animals have to reactivate that memory, and that also strengthens the specificity of that contextual representation. And by silencing the hippocampus during that period, maybe the brain was unable to selectively stabilize that memory, making that aversive experience more generalized and causing more generalization of fear. Now, therapeutically, this is quite important to think about as we think about developing treatments, that if we are selectively inhibiting, all of these cells, also cells that contain the specific memory of this trauma memory or this aversive experience, we might be increasing generalization. So we thought, okay, what if we selectively now just inhibit the ensemble of this neutral experience while the memory of the aversive experience continues to consolidate and become specific? Now, when we test them in the neutral event, in the neutral box, are we able to dissociate or unlink this memory? So here, Joe used another fancy molecular trick in collaboration with Steve Ramirez's lab. And he also decided that we're going to try both hippocampus and amygdala, two brain regions I previously told you is important for storing contextual fear memories. And so in this experiment, we can selectively tag just the cells that are being active as animals undergo exploration of this context A. And we can open this window when cells are active in that box, it tags those cells. And then when animals are done exploring context A, we can close this window by giving them doxycycline, this drug through food. And now only the cells are active around that time period are tagged. Now, Joe puts the animals back in the blue box, similar to many of the experiments I showed you before. And while the animals are in their home cage, he gives them this drug, clozapine, which in the HM4DI group will decrease activity of specifically the ensembles of A. And now he tests them for their behavior and see by inhibiting selectively the ensemble, can we get rid of linking? So in the control group, we find what I've previously shown you, which is that indeed the fear robustly, reliably transfers to this neutral context seen 2 days ago, despite never being shocked in that context. And this fear is, once again, selective, as animals do not transfer that fear to a novel context. Now, critically, what Joe was able to do was by inhibiting this ensemble during this period, he was able to unlink the two memories without increasing generalization. Future studies that Joe and team are currently working on now is checking out the hippocampus and amygdala separately, seeing their individual contributions to this process. Okay, so what they did here is that was to test the hypothesis that specifically inhibiting this ensemble is able to unlink these two memories during this offline period, and that might lead to the animals behaviorally separating these two memories, such as when they come back to this context, they are no longer triggering the aversive memory. So to bring it all home, the question I posed at the beginning of this conversation was, well, how do we link this aversive experience, such as here the example of my kids at the dentist, right, to past predictors so that we can make linked memories and make predictions about the future? So our working model, based upon the experiments I've shown you, as well as additional experiments I did not have time to show you, is that after an aversive experience, memories are encoded in a sparse neural population in the brain, And during some offline period, potentially during sleep, this is the hypothesis, although the data I've shown you, we did not record sleep, although those are current experiments underway now. But that during this offline period, not only is the aversive experience being reactivated, stabilizing, strengthening, being consolidated into long-term memory, but the more aversive the experience, it's almost like the brain is searching for past events that may be related to relate the two. And that by linking these experiences across time, the brain may be able to make predictions about the future and make better behavioral outcomes. And so we think that this might be, a circuit mechanism that underlies causal inference. Certainly not the only circuit mechanism, but one of. Now, in what I pitched to you so far, it seems like this is quite an adaptive a mechanism to link experiences in time to make predictions about the future. You can, another common intuition is that after, if you're sick, you might think back, ooh, what novel foods did I eat yesterday, and may choose to avoid that. And animals certainly do this in the wild. And making that association across long periods of time can be very predictive about not eating poisonous or food that may cause malaise. So while this is quite adaptive, I want us to think about cases in which this may become maladaptive. One of the things that I told you is that as we increase the negative valence, it increases the likelihood that past memories would be reactivated, linking the two. And in our model, we're not looking at trauma memories per se, but one of the future directions in the lab is to use mouse models of trauma and to see how pervasive is this linking, how extensive is it? Is this reactivation only within a day? Or does this somehow, after a traumatic experience, recapitulate nightmares and disturbed sleep that I think is common to PTSD? Does the reactivation of this aversive memory across long periods of time lead to upward linking and maladaptive linking, which may trigger fear and anxiety in the future? Those are speculations and ongoing work in the lab. So I want to finish with the take-home message of my talk today. So what I showed you today was that a lot of research in the literature has shown that memories are stored in neural ensembles distributed across the brain. It's not one brain region, but it's really across the whole brain. And a number of studies very recently have shown that memories can be raised or silenced or activated by manipulating these neural ensembles. And this is beginning to give us insight to think about, how can we manipulate trauma memories, drug addiction related memories, right? To understand about developing interventions and treatments. And the work that I showed you today that was directly funded by the generous support of Brain and Behavioral Research Foundation suggests that aversive experiences are linked with past memories during offline periods. And I think something broader suggests that the brain is quite dynamic. Memories are not like snapshots, but they continually update. And as we learn new information, we're integrating new information with their past memories so that we can make better decisions about the future. So to conclude, I would like to thank so much my amazing team. And honestly, this slide says it all. This is why I have the best job in the world. And every day I get to come in and work with amazing, creative, generous, kind, brilliant people who all care about the human condition and how to improve mental health. Actually, many of my lab members also volunteer and contribute to community service and working with patient populations as well. I've already highlighted the people who did the work, but once again, I want to highlight Joe Zaki, who is a very talented graduate student, co-mentored by Kanika Rajan, also Brain and Behavior Research Foundation award winner. I also want to highlight Taylor and Denise, Taylor Francisco and Denise Morales Rodriguez, who contributed to a lot of behavioral characterization, Zachary Pennington, who actually did a lot of the initial pilot work, helping us understand the different parameters, as well as test many of the chemogenetic and drug pharmacological manipulations. I also like to thank Phil Dong, who developed a lot of our calcium imaging analysis pipeline, in which I didn't get to go over today, but allowed us to be able to analyze that large scale population data that I showed you. Also, Zach Pennington developed all of the behavioral analysis pipeline that is also open source, which we use to characterize behavior. Really everyone in the lab contributed to the many discussions that led to the discoveries I presented today. And once again, I also want to thank my funding, particularly Brain and Behavior Research Foundation, who were one of the first foundations to invest in this high-risk project. And because of that early support, we were able to do the work and get a lot more funding to continue on this important work. And thank you so much for your attention. I will end with this take-home message, and we'll be very happy to take questions.
Well, Denise, I want to thank you for an excellent presentation, for the extraordinary work that you're doing. I love how you show just how it takes the whole team of people who are in the lab, more junior people in the lab, and more senior people over the years. For you, it really does take a team, and I appreciate you're also sharing the pictures and the stories about your family, which is really very, very, very nice.
Yeah, thank you. My mom said, tell everyone. I was like, Mom, is it okay if I share? She's like, tell everyone, if this works so well for me, I just want hope to be brought to everyone. So that was her message.
Great message. Thank you, Mom, on all of our behalf. So let me ask you, as you described, this is basic research in the lab with mice models, looking at very basic kind of information, looking at behaviors. How do you see this translating potentially to clinical care for people? Where do you see the next steps over the next period of time?
Yeah, thank you so much for that question. So, I hope I communicated that even though we are preclinical basic scientists doing mouse work, our questions are really translationally inspired. And, one of the things that we discovered is that the more aversive the experience, it changes how memories are reactivated. And it made us begin to wonder, in PTSD, for example, two of the cardinal symptoms of PTSD are increased nightmare and disturbed sleep. And this is long lasting. It persists months to years after the original trauma. And because of the technology that we've developed, being able to track the same cells across essentially the lifetime of the animal, we can then begin to model this in our mice. So what I showed you here is not a trauma model, but we have a mouse model where we induce acute trauma. So we shock the animal 10 times. And this leads to long-lasting changes in brain regions. This leads to increased generalized anxiety weeks to months later. It leads to increased startle responses. That is also related to some of the PTSD symptoms seen in humans. And so one of the questions that we're asking is, in this trauma model, after this acute trauma, how is this aversive memory being reactivated during sleep? And does it persist far longer? Does it cause to more linking across other memories across time that are unrelated? And if so, right, we can imagine how this might lead to other safe experiences, because now they're linked with this trauma memory. It might trigger, anxiety, fear, some of these negative responses. And furthermore, we want to know, can we intervene at later time points? So I showed you here, we can break the link after the initial period after the aversive learning, but we want to know, what is that critical window? Can we do this days later, weeks later? What are the boundary conditions for manipulating the spread of fear?
So it sounds like potentially this research would lead to a conclusion that the nightmares that occur in post-traumatic stress are not just the symptom, but also reinforce the trauma memory. So in some ways, if there's a way to decrease those nightmares, it not only would help with the current symptoms the person has, but maybe even help treat the actual post-traumatic stress disorder.
Exactly, that's our hypothesis. And one of the things that we're looking at now is there a specific sleep state in which this happens? Based upon my work from graduate school with Sarah Bendick, showing that REM sleep is really important for integrating your recent experience with past experiences. If that pans out to be true, such that it's a specific brain state, right? Then instead of what I showed you about selectively silencing ensembles, maybe by just manipulating or decreasing the amount of REM sleep, for example, we know that antidepressants and SSRIs actually decrease REM sleep. So there's already FDA-approved drugs out there that can target different sleep stages.
And is that work that you're looking at actually doing to test that hypothesis out?
Yeah, so right now what we're doing is we're recording both the neural activity and the sleep across long periods of time, so across weeks. And for us, it's important to model that across long periods of time, because in PTSD in humans, right, the nightmares and sleep disturbances are very long lasting. In the data I showed you today, this is like in the first hour after an aversive experience. But in human PTSD, the nightmares and sleep disturbances continue for months to years. And so we want to model that in mice and see if this persistence and reactivation of the trauma memory persists across, I guess in the mouse's lifetime is weeks to months, and if we can manipulate this pharmacologically with FDA-approved drugs.
In addition to post-traumatic stress, what other conditions do you see this type of approach potentially being helpful for?
Yeah, so another thing that my lab studies is aging. And so my prior work has shown that with aging, so, aged mice, middle-aged mice and middle-aged humans can, learn individual tasks or individual behaviors well, but they have a difficult time with flexibly updating. And this has been shown in both mice and in humans. And so we're using a kind of a similar approach, tracking the neural activity in aged animals and see how are they able to flexibly update their information. One of the things we also see with aging is that because they have trouble specifically temporally linking, so they tend to generalize your fear more, right? And so we think that that can be problematic if they're generalizing their fear more rather than being able to know specifically what they should be afraid of. And we are looking into, again, also FDA-approved drugs to manipulate the cellular properties and molecular properties in cells in aged animals to see if we can improve the specificity of the memory and the specificity of the linking so animals can update accordingly.
Great, thank you, thank you. One of the, and you saw the beautiful pictures of your kids, one of the concerns that we all have is the effect of the pandemic on children.
Yeah.
And if you look at a young child, a significant percentage of their life now has been in these last 2+ years of the pandemic.
Yeah.
How does this and the sort of the understanding of memory, do you see something there that may be helpful over the long haul as we see the impact of living through the pandemic on the younger generation?
Yeah, I think what I'm consistently amazed by is the resilience of young people, young animals. And so it's amazing how plastic and resilient their brain is. And I can even see it in my own kids. And now that they've been able to go back to school and have social interaction, I think one of the biggest stressors of the pandemic for our children, in addition to, of course, physical health, was the social isolation. And it's interesting because as a basic researcher, when we want to study stress, do you know what the most common way to induce stress or most reliable way to induce stress is by isolating our mice so that they can't play with their, friends or family. And that's basically what we've been doing to our kids over the last couple of years. And I definitely even see in my own kids how that has definitely exacerbated their stress, and they're much more sensitized and have negative responses to minor stressors, which is interesting because that is also something we model in the lab, and I can see it in my own kids. I actually recently went to my postdoc and I was like, oh my gosh, like what you see in your mice after acute trauma, and then you see this like sensitized response to minor stressors, way later is what I see in my kids now. So it'll be interesting to see, how the field comes up with ways to help our kids deal with that.
I think you're bringing up an extremely important point, and sort of broadening it from post-traumatic stress to just anxiety disorders, which we know were commonly occurring in children and adolescents, much more so than we had in years ago thought of, and now the effect of the pandemic. I'm wondering if, we know that talk therapy affects the brain physically, just as you were describing the research you're doing affecting mice brain. I'm wondering if there might be approaches in terms of talk therapy that may help physically affect the brain to help with anxiety and with memories associated with anxiety.
Right. I mean, so in the data that I showed you, right, that memories can be activated and that you can then maybe manipulate some of those trauma memories, but memories can also be activated by just recalling it, right? So as you're saying, in talk therapy, kids or patients can reactivate those memories, but now as they reactivate it over and over in a safe environment, they can dissociate maybe some of the negative experience that was associated with those experiences, and decrease the negative association and negative emotion with some of the experiences that our kids have had over time. I guess the big question now is how I've been trying to get my kids to therapy sessions, and everyone's booked. So we need more therapists. We need more healthcare workers in this field. We need more mental health researchers, because it's so important coming out of this pandemic.
I think you're right on target with that. One of the challenges is access to care for people due to the tremendous need and not enough treating professionals. Yeah, that's a very important issue.
I think in addition to access to care, if you don't mind me, Jeff, just saying, one of the amazing things that Brain and Behavior Research Foundation does is, in addition to providing funding, it helps educate, right, everyone about mental health. It helps break down the stigma. You created a space for me to share my story about my family, and I think that is a huge battle. So many people, one, are not aware, or even if they're aware, are ashamed to admit that they can get help, right? And so I think in addition to increasing access to care, we need to talk more about this. We need to break down the barriers and decrease the stigma. I think it's important that we all start talking more about this.
I agree with you, and my hat's off to you for sharing your family's experiences, to your mom for wanting you to share her experience, because that really does serve as a model for others to be comfortable seeking help, because we do have good treatments that help many people now. We obviously need research to get even better treatments to help more people, but you're right on target with that. Where do you see the research in your lab going 5, 10, 15 years from now? What do you see you and your lab doing at that point in time?
Yeah. So I started off my research actually working with human patients in sleep and memory, as I had alluded to, but I really wanted to understand the molecular mechanism, the cellular mechanism, right? What is really going on in the brain that underlies our memory system. And so I moved to doing mouse research because it gives us great access into the brain. But I think that as we move on into the future and as my lab dives deeper, and so in addition to looking at these circuits, because it's currently unethical to be able to genetically mutate human brain cells and manipulate circuits with this kind of precision and specificity, but if we can understand the molecules that may be different underlying trauma memories versus non-trauma memories, for example, then we can start developing, therapeutic interventions, pharmacological targets in which we can actually translate to human patients. And ultimately, what I see myself doing is, collaborating with clinicians and drug companies to move this forward in a more translationally relevant way.
Well, that is extremely exciting. And given your track record of success in what you've done thus far, I think we can, it's a pretty good bet that you'll be able to move forward with your goals as you pursue them.
Thank you so much. Thank you so much, Jeff.
Yes. So I want to, again, thank you, Denise, for the work you're doing, for taking time out of your schedule to share it with us. We will want to get an update from you in the not too distant future to hear more of what you've been able to do. So thank you very much. I also want to thank everybody that joined us today. And I would ask people, you heard from Denise the impact that the funding has on the science. So I would ask people to join us and consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientists. To make a gift, please visit bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you missed any portion of it or would like to share it with family or friends, please visit the events and webinars page on our website. And I want to invite you to join us again next month on May 10th at 2 p.m. Eastern Time, when Dr. Daniel Dickstein, Associate Chief and Director of Research in the Division of Child and Adolescent Psychiatry at McLean Hospital and Professor of Psychiatry at Harvard Medical School, will present Brain Behavior Alterations Underlying Self-Injury and Suicide Among Children and Adolescents. Thank you for joining us, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Once again, thank you. Take care.